All right, now to introduce a concept called anergy. Anergy means without ergy, or without ergo or zest for life. And that's what happens to immune cells sometimes. Why would that happen? Well, sometimes they can lose their stimulation and become non-reactive under certain circumstances. When would you want them to be non-reactive? How about if they are attacking your own cells, right? Right. That's when your body says, "No, you should not be able to react with my own cells, so stop it. I'm going to make you useless." That's what anergy is. Okay? Pretty straightforward. All right, so this slide is going to be a micro review. So, do you recall, back to microbiology, what toxins and what bacteria cause massive cytokine release by binding to the MHC receptor of macrophages and T cells, the T cell receptor, and complexing these two cells together, forcing them to interact and co-stimulate one another? So here's a toxin doing that. And thus, they release a massive cytokine storm and leads to systemic inflammation all over the body. What is that syndrome called? Toxic shock syndrome, right? Or toxic shock-like syndrome if it is. What bacteria cause those? Staph aureus and strep pyogenes. Very good. So definitely remember that from your microsections. What type of cytokines are we talking about here? Which ones cause the massive inflammation? We're talking like interleukin 1, interleukin 6, and TNF alpha. Okay? Also know the bacterial toxin endotoxin, which we talked about before. Again, and we just mentioned that this can bind to what receptor on macrophages? CD14, right? But this can bind to and stimulate macrophages to release cytokines, but also it causes what? What does it activate? What pathway? The complement pathway, right? And what specific type of complement? The alternative complement cascade. Very good. So now a brief word about antigenic variation. Now obviously, if you're a pathogen, the last thing you want to do is be detected by the body's immune system and killed off, right? So what do some of them actually do? Well, they actually change up some of the proteins on their surface. So for example, if it looked like this, they change it to look like this, so that immune cells become confused and don't recognize them anymore. That is through antigenic variation. And how exactly do they do this? Well, they can actually change their DNA. Remember with VDJ recombination, immune cells changing up their DNA to make different antibodies? Well, pathogens can do the same thing. They can splice out DNA segments and attach random segments together. So after it's transcribed, they can also splice out and rearrange their RNA segments too to make unique cell surface proteins. And then, what's the purpose of that? Then they can be undetected by your body's immune system. Kind of sneaky, right? So what organisms do this? Well, I'm not going to go through all of them, but just think of Salmonella with its flagella, Neisseria with its pili, Borrelia, and even some viruses like the flu with H1N1, and trypanosomes. That's a protozoa, so that can do it too. All right, the concept of passive versus active immunity. So we talked about this earlier when we mentioned that when infants, they're not able to make their own antibodies yet, so mothers actually help them by giving them antibodies. They even gave them to them in the womb through the placenta. They also give them after birth through what? Through colostrum, through breast milk. And they're given what type of antibody specifically? IgA, right? Okay, so essentially the main difference between passive and active is, are your immune cells being passive or are they being active? So if they're active, then your own immune cells are actually being stimulated and making antibodies themselves. And this leads to a more robust immune response and a greater memory to pathogens. It makes sense because if you think about it, when the pathogen is processed by your own cells, like we see here, presented on the surface, and your own T cells are stimulated, they'll release cytokines that will lead to memory formation and a more robust response the next time you're exposed. This won't happen if you're just passively given antibodies. You won't know what the heck you're reacting against because they're not your antibodies to begin with, right? So we just said that when you have active immunity taking place, the next time you're exposed to that pathogen, you have a robust response. So wouldn't it make sense then to mimic a pathogen so that when you are exposed to the real pathogen, you have a strong response to it, right? Well, that's what vaccines are, okay? And there are two main types. There are live attenuated vaccines that are like the real live form of pathogen, but they're just weakened. And those involve, that's the closest thing to the real pathogen as you can get. So you consume your immune cells, or your normal cells consume the pathogen or the pathogen-like substance, and it gets processed the same way as a pathogen would, presented on the surface just as we saw before. And your immune cells, your T cells, are stimulated to the max, fully stimulated, release cytokines that induce the best memory. Okay? Another type of vaccination is inactivated vaccines. So these are actually not living at all. And this is close but not very similar to the real thing. This is just when we take a little part of a pathogen, heat up a bacterium or something, and take a fragment from that bacterium and expose your cells to this. So it's not the full thing because it's not degraded properly like we saw before, and so we don't have quite as good of a response. But this will stimulate B cells still. It'll still stick on the surface of B cells to cause them to make antibodies, right? So you'll have a lot of antibodies around, but you won't have the strong memory response that you would have had if T cells were involved. You need T cells for memory. All right, it's about that time again for a flash quiz. So, what are examples of killed vaccines? Well, you can remember them with the mnemonic RIP always, because they will stay dead. So they are rabies, influenza, and polio, as well as hepatitis A. Hypersensitivity time. Time to review hypersensitivity. So sometimes your body becomes overly sensitive to things, to the point where it actually causes harm. So there are four main ways in which this can happen, and you can remember them with the mnemonic ACID. So the A stands for allergic, and that's type 1. Type 2 is cytotoxic. Type 3, immune complex. And type 4, delayed. So we're going to talk about all of these in great detail right now. So we'll talk about the A first, or type 1. And we actually already mentioned this in great detail before. So this is the allergic response that we see with like bee stings and other types of anaphylaxis, from like dust and pollen. So type 1, allergic, okay? So remember we had B cells. Here's a B cell. And after it class switched from it was making IgG, something caused it to class switch to make IgE. What cytokines or what interleukins cause that? Remember from T cells, interleukin 4 and 5, right? Hot T-bone steak, remember that mnemonic, right? Yes, this caused the B cell to class switch. By the way, what receptor is needed? CD40 and CD40 ligand, right? Okay, good, just making sure you're paying attention. So the class switching to make IgE. So this is after the first exposure to an allergen, like pollen or something. And then this IgE will go and stick on the surface of mast cells so that the second time you're exposed to this allergen, you have cross-linking and massive degranulation of the mast cells. What do they contain? What do they release? Histamine, bradykinin, other things that you'll learn about in the inflammation section, which is the next chapter. So that just leads to massive inflammation and potentially anaphylaxis if there is enough allergens. The C is for type 2 hypersensitivity, cytotoxic hypersensitivity. And you can think of cytotoxic, actually, to think of type 2. So it's toxic to cells, right? Right. And what is the main mediator of this? Antibodies, actually. So remember, antibodies connect to antigens, right? The word connect, the first three letters, helps you remember what antibodies do. So when antibodies attack your own cells, they can activate complement, as we discussed earlier. They can opsonize those cells, like we talked about, remember? It's like giving the macrophages something to latch onto so it can consume your cell. Or neutralize cells, like we talked about before. So those are the three main things. There's another thing too called antibody-dependent cell-mediated cytotoxicity, or ADCC. And this is basically natural killer cells destroying cells that have bound antibodies. Through what mechanism? Apoptosis, right? So here's an antibody bound to your own cell, natural killer cells. What do they contain and what do they release? These are called granzymes, right? Simulate apoptosis. So type 2 is primarily antibody-mediated attack of your own cells. I is for immune complex, and this represents type 3. And you can think of three things involved. So there's an antibody, an antigen, and then what is this going to activate? The classical complement cascade. So three things involved with type 3. That's a good way to remember it. This antigen and antibody complex, what's that called? An immune complex. That's another name for it. So this antigen antibody immune complex is going to be traveling through your bloodstream and eventually in small arteries, like in your peripheral, fingers and toes, and other small vessels, maybe in your kidney. These immune complexes will get stuck, and when they're held up, there will be enough time for complement to be activated. Okay? And that's not good because C5a will be activated and called in. And what does C5a do? What is that chemotactic to? Neutrophils, right? Neutrophils. Oh no, what do they release? They are non-specific killers by releasing reactive oxygen species, and this will tear up your tissue. Just destroy everything in sight. And that's why we have kidney disease and other diseases of small vessels in people with immune complex type 3 hypersensitivities. We'll talk about some examples in a little bit. But basically what you need to know now is if it's systemic, if they're traveling through your whole body, that is called serum sickness. If it's just a localized reaction, that is called the Arthus reaction. All right, now it's time for the last hypersensitivity, and that is the D of acid for delayed type 4, okay? And the reason it's delayed is because it doesn't involve antibodies. So antibodies are actually very fast acting. But this, type 4, the only type that does not involve antibodies, primarily involves T cells, so it's a little bit slower. So this is what we see, for example, in poison ivy, when you brush past some poison ivy on accident. There's this antigen, do you know what the name of it is? It's the rhesus antigen. It is deposited on your skin. What are like the macrophages of your skin? The Langerhans cells or the dendritic cells. So they're going to process this rhesus antigen, present it on their MHC what receptor? Nice review. MHC2 receptors. Complex with what other type of cell? T cells. Okay, specifically Th helper type 1 cell. All these receptors will bind. You'll have the first stimulation, is this B7 CD28 complex. And the second stimulation, as we know, immune cells always need two stimulations to be activated. The second one of which is involving the cytokines interferon gamma from the T cell and what from the Langerhans cell? TNF alpha. So this co-stimulates both cells, and we have activation, stimulation, and memory to this rhesus antigen. So that the next time you're exposed to this poison ivy, unfortunately, you will have a pretty bad immune response in that rash that we always see whenever we walk in the woods without looking around. Now for some specific types of hypersensitivities. We'll just mention them briefly because you'll learn a lot more later on in other sections. But for example, type 1, we just said that, anaphylaxis was an example, bee stings, allergies, other types of allergies to pollen and dust mite feces. Okay, so that's type 1. Type 2 was cytotoxic, and examples of this, can you think of some? Are usually localized autoimmune diseases, so like rheumatic fever in the heart, when antibodies attack what valve is typically involved? The mitral valve, that's type 2. When your thyroid is attacked or stimulated, actually, in what disease? Graves' disease. So anytime you have antibodies attacking your own cells, there's usually a localized autoimmune reaction. Type 3, remember 3 things stuck together: antibody, antigen, and then complement activated. So the best example is in systemic lupus erythematosus, or lupus for short. Has all kinds of systemic diseases, and you'll learn a lot more about that later. Type 4 is delayed, and where do we see this? We just said with poison ivy, but all the contact dermatitis reactions and the reactions that are delayed, as its name implies, delayed type hypersensitivity. So poison ivy, like we said, also the PPD skin test is type 4 hypersensitivity. So even though we try to prevent them, sometimes we still have blood transfusion reactions. There are 4 main types that you should know. We'll talk about each one of them briefly. The first one is just a mild allergic reaction. And recall the mechanism of allergy. What type of hypersensitivity? Type 1, involving mast cells with IgE on their surface. And the second time they're exposed to an allergen, like potentially the donor has some pollen in their blood that the recipient is allergic to, okay? And so they just have a mild allergic reaction. Usually you can give them some Benadryl and they'll be okay, because there aren't too many of these allergens in their blood. But if, for example, let's say a person is IgA deficient, they have a decrease in IgA antibodies. Some people have that. And so when they're given blood that contains a lot of IgA, they go into anaphylaxis. All right, and that is not good because there's just massive amounts of IgA. So whenever there is a great quantity of allergens in the blood that is being donated, the recipient could potentially go into anaphylactic shock due to the histamine released from all these mast cells being degranulated. So that's not good. The next type is febrile non-hemolytic transfusion reaction. What do you think this is due to? Well, this is, just as it implied, it's non-hemolytic, but it's due to antibodies in the recipient's blood that bind to immune cells from the donor's blood. So what is this going to activate? Complement cascade, right? Right. So this activates complement. C5a will be around, and also because these antibodies are binding to white cells, these white cells will release cytokines. So what would cause a fever? What would make someone febrile? What interleukin? Interleukin 1. Very good. Remember, it releases prostaglandin E2 in the hypothalamus. So that's that one. The last one is the bad one, acute hemolytic transfusion reaction. And this is just when someone wasn't careful and didn't see the person's blood type, did not cross-match the person's blood type appropriately. So this is bad. This is when antibodies, which you have against every type of blood that's foreign to you, they react against the foreign blood cells and leads to massive inflammation and a lot of problems. That person, you need to stop that transfusion immediately and monitor them symptomatically and hope that they don't crash. So autoantibodies, speaking of type 2 hypersensitivity, this is just going to be an overview slide and I'm going to quiz you. So basically, can you think of any autoantibodies associated with the following diseases? The first one we'll talk about is rheumatoid arthritis. Do you know any off the top of your head? So you'll learn about this later, but it's anti-CCP is one and rheumatoid factor is another. Okay? Both of those are antibodies associated with that. What about SLE? What antibodies are associated with this? So a bunch of them, but some of the ones you should know, well, obviously anti-nuclear antibodies associated with that. There's others that are specific to it, like anti-double-stranded DNA and anti-Smith. So know those ones. Scleroderma, anti-centromere antibody, and SCL70 antibody. So those antibodies help you diagnose and confirm that a person has a disease. There's others as well, but I'm not going to get into them. You're going to learn about them in the later sections of the book, and you can review them on that page as well.